So today's topic of discussion is enzymes. Enzymes are biological catalysts that can increase the rate of a reaction without changing the equilibrium. That means they increase the rate of forward reaction as well as rate of backward reaction, but the equilibrium is not changed. Means the enzymes do not change the delta G value of the reaction. The delta G, which was without the enzyme, remains the same even after activity of the enzyme. It just increases the rate of the reaction. Next, a rate of the reaction V is directly proportional to the transition state of the substrate S asterisk. Every enzyme substrate reaction, the substrate remains in a ground state. When it is converted into a product, it is first converted into a transition state. This state is the transition state S asterisk, from where it is broken down to the product, and the difference is called the delta G. The free energy difference of the substrate and the product is called delta G. So this y-axis is actually free energy. Free energy. So how can this can be achieved? The V can be directly proportional to transition state, concentration of the transition state of the substrate. How the concentration of the transition state can be increased? There are two ways to do that. One is by heating and another is by adding enzymes. Heating will increase the Brownian motion of the molecule, and that is why the reaction will be faster, but there is a limit. After a certain temperature, the process becomes inhibited actually, and there is a Q10 temperature coefficient also called, that at every 10 degrees centigrade rise of temperature the rate of reaction doubles, but there is a limit. It does not cross 50 degrees centigrade in mesophilic organisms. So coming to the mechanism, how enzymes actually increase the transition state of the concentration of the transition state of the substrate. How do enzymes increase the rate of reaction? This is actually free energy. This is delta G. An uncatalyzed reaction like this one, yellow one, you can see this is activation energy EA of uncatalyzed reaction. It is very high. When enzyme is added, this value, this is the EA of catalyzed reaction, cat. This is uncat, you can say. So the activation energy EA of a catalyzed reaction is very lower than an uncatalyzed reaction, and that is why the substrate is quickly reaches to the transition state, wherefrom it can be broken down to the product, delta G remaining the same. So that is how enzyme brings about a reaction faster. But how does enzyme decrease this EA level? The question is that the EA of a reaction. How does enzyme decrease the EA of a reaction? The enzyme does so. The enzyme lowers the EA value. From it takes the binding energy to lower the EA value. Binding energy, what is that energy? This energy is basically the interaction energy between the enzyme and the substrate. When the enzyme and substrate interact, there are so many bonds are being formed, like the hydrogen bonds, van der Waals interaction, hydrophobic interactions, maybe ionic interaction also. So all these weak interactions, they generate energy that is called binding energy, and that energy is basically required to lower the EA of a reaction. Now coming to how enzyme and substrate interact together. There are two theories regarding this. The first is the lock and key hypothesis, as you already know. Another is induced pit hypothesis theory. Lock and key hypothesis was proposed by Emil Fischer way back in 1890s, whereas induced pit was proposed by Koshland in 1966. The lock and key hypothesis is basically says that all enzymes have a specific substrate, like a lock has a specific key. So enzymes will bind with specific substrate. It has an active site that exactly fits with the substrate, and then the enzyme substrate intermediate is formed, which is later broken down to enzyme. Enzyme is released and product, the substrate is converted into product. The induced pit hypothesis says that the enzyme active site is initially not exactly same as the interacting sites of the substrate, but when the substrate is allowed to interact, the active site is induced to change its conformation so that it fits good with the substrate. But the problem with these theories lies in the fact that if an enzyme has a exactly fitting site with the substrate, then it will form a structure like this. Enzyme substrate, this will form a structure very stable and which will have an energy level much lower than in fact without the enzyme uncatalyzed reaction. So if the energy of the ES goes down this level, then the EA value will further increase and it will become impossible for the enzyme substrate to be converted into product. So literally this theory would not explain how enzyme substrate interaction takes place. A little modification of the theory is that the specific site, the active site of the enzyme, is exactly fitting with the transition state of the substrate, not with the normal state. So this gap here is because this when the substrate is converted into a bent structure like this, so the enzyme's active site is fitting well with this transition state of the substrate, not with this normal state. And when the transition state is achieved, the substrate is in strain and it is immediately broken down into a product. This is an unstable condition, and that is why when enzyme is added, the ES level, the energy of the ES level is a little bit higher than uncatalyzed reaction, and it can easily achieve the EA, activation energy level of the reaction. This is how the interaction between enzyme and substrate occurs actually. Now let us see the properties of enzymes. As already mentioned, enzymes are very substrate specific. Each enzyme has a specific substrate. No enzyme can bind with different substrate which it is not actually supposed to. Enzymes are catalytic, they can increase the rate of reaction, but they do not change the free energy of the reaction. Proteinaceous, enzymes are always, not always, 99% of cases they are proteins, because it has been found if you increase the temperature beyond 50 degrees centigrade, maybe 55 degrees centigrade for some time, most enzymes get denatured and become inactive. But there are certain cases like ribozymes, RNA acting as enzymes or catalyst. This is called ribozyme. Such examples are found in case of 23S rRNA of protein synthesis, and also in case of group 1 and group 2 splicing introns. Those are ribozymes basically. Uh, Enzymes are also reusable, and they actually are involved in, most of them are involved in reversible reaction. They are again available for the next round of reaction, but every enzyme has a particular half-life. So these are the basic characteristics of enzymes. And enzyme kinetics. Now, as we know, if we do a reaction in which the concentration of the enzyme is constant, enzyme is remaining constant, but the substrate is gradually being increased. You are adding increased amount of substrate as time goes on. So what you will see how the velocity of the reaction will behave. Initially, when the substrate level is very low, the rate of reaction will be also low. When you are adding substrate concentration, increasing substrate concentration, rate of reaction will increase like this. It will be linearly increasing to some extent. And when the substrate concentration is nearing saturation, the velocity will become constant. This is the maximum velocity, Vmax, that an enzyme can achieve. This is because the enzyme concentration is constant, and every enzyme has a specific active site. Say there are 10 enzymes with 10 active sites. So when you are increasing the concentration of the substrate, 1 substrate, 2 substrate, 3 substrate, 4 substrate, and so on, till it reaches 9 and 10 substrate, the velocity is increasing. But after you have added 10 substrates, no further site is available to bind with, and so the reaction becomes constant. So this is the Vmax value, and this is the half Vmax, half of the Vmax value. So at half Vmax, the enzyme concentration, the substrate concentration is this one. This substrate concentration, which is present at the when the velocity is half the maximum, is known as Km value of the enzyme. Km is the Michaelis-Menten constant, and this whole reaction could be expressed by Michaelis-Menten equation, which is V0 equal to Vmax into concentration of the substrate divided by Km plus concentration of the substrate. Now Km is what? Km is the substrate concentration, this substrate concentration, when the velocity is half its maximum value. So Km is equal to S when V0 is equal to half the Vmax. And this Km value defines the activity or kinetics of an enzyme, how it will behave. Less the Km value of an enzyme, more active is the enzyme. For example, say an enzyme E1 has a Km value of say 0.5 micromole. An enzyme 2 has a Km value of say 1 micromole. Which enzyme is more active? This enzyme, because it reaches its half maximum value very early when the substrate concentration is merely 0.5 micromolar, whereas this enzyme reaches the same level when the substrate concentration is just double of this one. That is why this enzyme should be said to be slower. Now, factors affecting enzymes. The most important factor is the substrate. Just mentioned a little while ago that when you increase the substrate concentration, the rate of reaction increases, but it comes to a saturation level. This curve is called substrate saturation curve. This is Vmax of the substrate, and this is the Km value of this reaction. So substrate affect the rate of reaction. Enzymes are directly proportional to the concentration. If you have kept the substrate concentration constant and you are continuously increasing the enzyme concentration, then the rate of reaction will increase continuously, and so on. pH, this is very very important. Every reaction has an optimum pH level, and this would be in case of microorganism, even in case of human beings. It depends on the tissue where the enzyme is. Enzymes present in the stomach act at very very low pH, and those which are present in pancreas have a high pH requirement. But normally most of the enzymes have a pH requirement of 7. That is the optimum value. When pH is increased beyond that, the activity quickly falls, and at the down at lower pH also, say at the level of 5, the activity is also very poor. This is pH. Same is the case with the temperature. Temperature should also be optimum, and that also depend upon the type of organism, where is it growing. If an organism is growing in hot water, if it is a microorganism of sulfur spring, then the temperature on ambient temperature will be higher, 70 degrees centigrade or so on. And if it is a psychrophilic bacteria, then the ambient temperature will be lower, below 5 degrees centigrade, or between 5 to 15 degrees centigrade or so on. But most organisms that are mesophilic, they have a 30 degrees centigrade requirement of optimum temperature for all enzymes activity to be maximum. So if you increase, according to Q10, if you increase 10 degrees centigrade temperature, the rate of reaction will double, but beyond 50 degrees centigrade, it will not result in the same way. The enzymes will get denatured because of its proteinaceous nature. Oxygen is very very influential for enzymes, because oxygens might be converted into superoxide radicals, hydrogen peroxide, and also hydroxy free radicals. These are all called reactive oxygen species, and they can bring huge changes in the structure and property of proteins and enzymes, membranes, nucleic acids, and all macromolecules. Enzymes like nitrogenase is well known to be inhibited in presence of oxygen. So because the disulfide linkages that are present, when they are reduced, they are converted into SH thio groups, but when they are oxidized, they are converted again into disulfide linkage. In certain enzymes, this condition will be inactive condition, and this will be active condition. So presence of oxygen is important for enzyme activity. There are certain enzymes which have two sites. One is substrate binding site, and another is modulator binding site. Modulators could be of two types. Either it will be inhibitor or it will be activator. Activator will increase the rate of reaction. Inhibitor, as the name suggests, it will inhibit. Such enzymes having two sites are called allosteric enzymes, and they behave a little bit differently. They have a sigmoid curve like this. When you increase the substrate concentration, the velocity of reaction will behave like that. Against the normal, this kind of curve, it shows a sigmoid curve like this. These are allosteric enzymes. Now about the concept of cofactors, coenzymes, and prosthetic groups. An enzyme having cofactors or coenzymes is together called holoenzyme. Holo means complete. It can be broken into two parts, proteinaceous part and non-proteinaceous part. Proteinaceous part is called apoenzyme, non-proteinaceous part is called cofactor. Cofactors could be of two types, inorganic cofactors, organic cofactors. Organic cofactors are again of two types, strongly bound, those are called prosthetic groups, and weakly bound, those are called coenzymes. So this is the example. For example, if you take an example, a prosthetic group like pyridoxal phosphate is a prosthetic group. TPP, thiamine pyrophosphate, is a prosthetic group. Lipoic acid is also a prosthetic group. but like FAD is a coenzyme. Maybe in certain cases NAD is a coenzyme. Coenzyme A is a coenzyme. So these are the coenzymes which are weakly bound with the substrate, and these are directly covalently linked with the enzyme. Those are called prosthetic groups. Now there are few terms like isozymes, abzymes, etc. Isozymes are basically different forms of an enzyme that can catalyze the same reaction, but can be inhibited by different inhibitors. These are actually the enzymes of branched pathway. Suppose this is a substrate that is converted into two products A and B, but the enzyme required is one, E1. So E1 can be inhibited by, E1 has two forms, say. One form will be inhibited by A, feedback inhibition. Another form, maybe E1 prime, will be inhibited by B. So E1 and E1 prime, these are the two forms of the enzyme E1. Those are isozyme. They actually convert substrate into an intermediate product, this, and that ultimately is finally converted into A and B. So this reaction is common for both of them. So they actually catalyze the same reaction, but can be inhibited by different feedback reactions with different products. So isozymes are the isoforms of same enzyme. Their molecular weight might be different. If you run a gel, then you get different band position for each of the isozymes. Abzymes. Abzymes are antibodies that has been converted or modified into enzymes, and this has been done to increase the specificity of enzyme and also to understand the enzyme substrate transition state, because that is very unstable. But if an antibody will behave like an enzyme and will bind with its substrate, the structure will be stable. That can be studied by X-ray diffraction, NMR, and other molecular techniques.